Welcome, everyone. My name's Liz Myring. I'm the president of the Protein Society, and it's my great pleasure to be introducing this exciting webinar today. I'm going to start with a few general announcements before the presentations from our speakers, which you can see here. First of all, this webinar is part of an ongoing series. Our next webinar is January 31st, Protein Engineering for Biotechnology and Enzyme Catalysis. And I just want to invite everyone to make suggestions for topics and to consider organizing one of these webinars yourself. This is really something we're doing for our community, and Pernilla Wittung-Stafshede suggested this topic, which I'm thrilled that we're presenting today. So our staff at the Society make this really easy to do. Your focus is on the science. So you can contact me or Raluca Kedar, our CEO, and our information for the webinars is on the Protein Society webpage. So I wanted to also just briefly mention that we have our annual symposium this next year in July of 2024 in Vancouver, Canada. So I encourage you to look at the program. It's also available on our webpage, and I hope to see many people there. And I'd like to also note another thing that our society does is have this Journal of Protein Science. So I encourage you to send your papers there. We have special issues as well, which you might want to consider. You can also suggest topics for that. And without further ado, I'd like to come to our speakers for today. So this webinar was kindly organized by Pernilla Wittung-Stafshede. Thank you very much, Pernilla, for doing this. And I'd also like to thank our speakers, Matt Chapman, Ehud Gazit, and Margaret Sunde. We're going to hear from our speakers in the order that you see here on the slide. First, Pernilla. So this topic of biological amyloids is an exciting one because amyloids are increasingly being discovered to have a variety of roles, both functional roles for the organisms in which they exist and roles in disease. So we're going to hear about a breadth of topics here, and they're being engineered. So I don't yet know the details. I'm looking forward to the secrets that we're going to hear about today. So Pernilla will start with catalytic activity of amyloid fibers. Then Ehud Gazit will talk about metabolite and protein amyloids from newborn metabolic disorders to age-related neurodegeneration. Then Margie will follow on with roles for functional amyloids on either side of the host-microbe divide. And Matt Chapman will be our final speaker, bacterial functional amyloids and the link to neurodegeneration. So before I turn this over to Pernilla, I just want a couple of logistical notes for you all. So each speaker will give their presentation, followed by a question and answer period. So you can at any point type questions that you have in the chat. So look for the little chat icon. The questions, when you type them in, will be visible only to the organizers, not to everyone. And then we'll take questions at the end of the talks, and perhaps also at the very end of all 4 talks, depending on time. Some of the questions will be answered orally, so we'll read out your questions in the question period, and some may be also answered in the chat, so there may be typed answers, and once those are answered, either orally or in typed form, they will go into the answered section and you can all see them. So with that, I'd like to thank everyone involved in this, also Raluca Kedar and Shannon Cunniff, who are our staff who make this work so well. And I'd like to thank Pernilla again, and Matt and Ehud and Margie. And I'll turn it over to you, Pernilla. Thank you.
Thank you very much. Oops.
I need to stop sharing. Hang on.
Oh, yeah. So, should we get rid of that picture?
Yeah, I'm trying to stop sharing.
Oh, now I'm big. Okay. So I also want to say welcome to everyone that's here. We can't see you out there, but I hope there's a lot of people there. So first, thank you, Liz, for inviting me to do this. We happened to meet at a live meeting this summer, you and I, and then you pitched this idea, and then I followed up. And to everybody that are thinking about something, it was really easy. I can confirm that. You just gather a bunch of people that do some interesting research in an area and you propose that to the Protein Society. So, I think this is a great complement to real live meetings where we meet each other, but this is another format where we, still can share our results, but in a much bigger scale over the whole world. So we have 4 speakers with me today, and we're sitting in Australia, in Israel, in Sweden, and in the US. So we have very different time zones. Some of us are up very early, some of us are about to go to bed. So, and that's probably the same for you out there. So thank you to the Protein Society. Thank you to my co-speakers here that you agreed to do this. The 4 of us together, I think that makes this unique and exciting to listen to. I look forward to hear my co-speakers' talks and what will come out of there and the discussions that follow. So like Liz said, please ask questions, put them in the question and answer, and we will try to answer a lot of questions and have a discussion. So the webinar title is Emerging Functions of Biological Amyloids, and you will hear 4 different aspects of that from us here now. And I'm not going to say so much, but we'll talk about human amyloids, we'll talk about functional amyloids in bacteria, in microbes. We talk about tiny amyloids, the smallest you can even imagine. So you'll get an array of results, I think, or interesting findings that I think is important to think about. Many people have studied amyloids, mostly for the mechanism of forming them, but this about what kind of functions do they have? That's kind of a newer way to look at things, and there are more things to discuss. So with that, I think I will start. And I will go first, so I will introduce myself, I guess, by sharing my screen. So let's see, probably everybody is turned off, but now does it look good? Okay, if it doesn't look good, somebody says that. So I will start out by talking about, oh, somebody raised their hand. Ooh.
No, you're good.
Okay, I'm good. Oh yeah, that's of course. It was actually some time ago as I did a Zoom webinar or a Zoom seminar. So it feels like I forgot all the details here, but now we're fine. So I will talk about catalytic activity of amyloid fibers. So I'm Pernilla, I'm a professor at Chalmers University of Technology in Sweden, in Gothenburg. So you all know that most proteins need to fold to compact structures in order to function. That's a central dogma. But many proteins we know of today are also intrinsically disordered in their functional states. But regardless, if you have problems with protein folding or misfolding to the functional state, that can lead to human diseases. And we can have loss of function type of diseases where the folded protein is not made properly and you lose that function. But you also have gain of function type of diseases involving amyloids. We call them amyloid disease. Here, proteins misfold and start to aggregate into ordered fibers or plaque with this beautiful cross beta sheet structure where you have beta strands lined on top of each other. Amyloids are involved in at least 50 human diseases that we know of, probably more. And the most famous ones are Alzheimer's, Parkinson's, Huntington's, type 2 diabetes, prion diseases, ALS. Yeah, and I had a picture. These amyloid fibers are long, so you can also see them under the microscope, and you see how thin they are and how long they are. But I want to point out that these diseases, especially neurodegenerative disorders like Alzheimer's and Parkinson's, is a huge problem in society. Age is the largest risk factor, and the population in the world is getting older. And that's good, right? We all want to live longer, but we want a healthy life. And for these diseases, we have no cures. We can only treat symptoms for a while. So it's predicted to be just a bigger and bigger, healthcare problem in society in the future. So in my research group, we focus on the protein involved in Parkinson's. And this protein is called alpha-synuclein. It's an 140 residue, intrinsically unstructured protein. It's involved in neurotransmission, synaptic vesicle transport, something. It's actually found in the whole body, but it probably has functions in the brain too. It has a membrane binding part in the N-terminus, and when it binds to membranes, it adopts a helical structure in that part. Then the N-terminus is positively charged, the C-terminus is negatively charged. So this peptide can assemble into amyloid fibers and look similar to the amyloid fibers found in these other diseases I mentioned, where other proteins would assemble to those amyloids. And the hallmark of Parkinson's disease is something called Lewy bodies. They are found in dopamine neurons in the brain, in the substantia nigra part of the brain in patients. And these Lewy bodies contain alpha-synuclein amyloids, and that's how the protein was linked to the disease in the first place. And it's still not clear what species along the progression from monomers to amyloid fibers that are most toxic and actually cause disease. Maybe smaller assemblies, oligomers, are most dangerous. So we take biophysical approaches mostly to learn more about alpha-synuclein's cross-reactivity with other proteins, other molecules, mechanisms of these conformational changes, and mechanisms of assembly status, and so on. So over the maybe decade that we have studied alpha-synuclein in my lab, we've identified many players. We looked at synthetic small molecules, how they can modulate the aggregation reaction, and we can make through them concentrate intermediates that we can study. We looked at crowding effects on this reaction. We looked at cross-reactivity between other human amyloid proteins and synuclein because they can trigger each other sometimes or block each other. We look at mechanistic aspects of assembly, copper ions and copper homeostasis and copper transport proteins because alpha-synuclein can bind metal ions. We also looked at perturbations of DNA because synuclein seems to bind DNA and can stretch the DNA, and that might be related to gene regulation. We also looked at bacterial proteins from the gut, from the microbiome, how that can affect aggregation, membrane interactions. I said it was a membrane binding protein. We also found a fish protein in food that can affect aggregation of alpha-synuclein. So most of the things we have done is really focusing on molecular mechanisms and external modulators of this kind of assembly process. What I will talk about today now is focusing on the amyloids, the final product here, the amyloid fibers, because we have found that they seem to act like enzymes and do chemistry. And this might be a new piece to this puzzle that's already a complicated puzzle of amyloid-related diseases. So, can amyloid fibers really do chemistry? Well, amyloids are considered chemically inert species. Yes, they can transfer from cell to cell. They can promote new fibers through elongation and secondary nucleation processes, so you can make more fibers, so they're definitely bad. But that they can do chemical reactions or catalyze chemical reactions, nobody thought of before. But amyloids, the amyloid scaffold had also been exploited in materials research. And people have designed synthetic peptides that assemble into amyloids that display catalytic sites on the surface. For example, a metal binding site that's protruding from the surface or a fusion protein. And then they can do catalysis. But it wasn't until a few years ago that somebody showed that natural amyloids, these human amyloid proteins as they are, in the amyloid state could catalyze chemical reactions. And this was shown in two papers in 21 and 22 by the group of Rassi Elenik in Israel, that he showed that amyloid beta amyloids, this is a peptide in Alzheimer's, it could catalyze ester bond hydrolysis and oxidation of dopamine. And then in the second paper, he showed that glucagon amyloids, glucagon is a hormone that we have in our body, those amyloids could also catalyze ester bond hydrolysis and dephosphorylation. So when I saw this, I thought maybe this is more general than we think. We need to test this on alpha-synuclein amyloids. So that's what we did. So first we tested esterase activity. And here we used a model compound or model substrate, paranitrophenyl acetate. So when you cleave the ester bond, you get a yellow product that you can measure by spectroscopy. And we made alpha-synuclein amyloids in a two-step process to try to make as homogeneous samples of amyloids as we could. So when we test this substrate with synuclein amyloids, what we find is that we detect ester hydrolysis for the amyloid samples, but not for synuclein monomers when I have the same amount of monomers as I have peptides in the amyloids. If I truncate synuclein by 21 residues in the C-terminus, so I chop off some of that C-terminus, or if I mutate the sole histidine in synuclein to an alanine, that doesn't matter. We get the same esterase activity for the amyloid form. Okay, so then we tested dephosphorylation as well. Here we used a similar model substrate, but now a paranitrophenyl phosphate. So if you cleave the phosphate bond now, you get a yellow product. So again, we see this dephosphorylation in presence of the amyloids, but not for the monomers. And here again, truncation doesn't matter. That C-terminus doesn't seem too important for this. But when I now remove the histidine and substitute it for an alanine, I diminish amyloid activity. So the histidine is important for phosphorylation or dephosphorylation. And actually, we followed up on this a little bit more recently, where we took ATP instead of a model substrate, and we incubated amyloids and monomers with ATP, and then we used HPLC to separate the products. And what we find after a few hours of incubation, that in presence of amyloids, we almost convert all the ATP to AMP. So we seem to get rid of two phosphates. But the monomer, as you can see here, doesn't do a thing to the ATP that we had in the sample. Okay, so I showed you catalytic activity, but what's the magnitude of the effect? So we analyze the data for the model substrates, and we can do Michaelis-Menten analysis and get these Km and Kcat parameters. So here I show you the parameters for the 3 different synucleins that I tested, and I also compare to the amyloid beta amyloids and the glucagon amyloids from the 2 papers I mentioned. So you can see that amyloid beta and glucagon amyloids, the activity that was reported for them, for esterase and phosphatase activity, is very similar to what I see for alpha-synuclein amyloids. You know, Km in the millimolar and Kcat in something 0.0 something, or maybe 0.00 for A beta and glucagon actually. So synuclein is faster. If I compare this data to real enzymes, of course, they are much better. I found some values for real enzymes doing esterase and phosphatase activity, and they are orders of magnitude faster. But these enzymes have evolved, right, to do this duty only. If I compare to designed catalytic amyloids, that people have made peptides to do esterase activity, they fall in the same ballpark as alpha-synuclein amyloids by themselves. So, you know, natural alpha-synuclein amyloids are as good as, you know, whatever people have designed to do esterase activity. So how can an amyloid fiber catalyze reactions? Well, if I look at an amyloid structure or alpha-synuclein amyloid, so here I'll show you one structure. There are many structures reported, and they have variations depending on the exact conditions in this beta sheet core of the amyloid structure. But then you have the floppy N and C termini. But if you look at these surfaces, you see that there's many putative active sites. You have charged residues, you have catalytic triads maybe at different positions. The histidine lines up on the outside, but you have these repetitive motifs on the surface because the beta strands are packed on top of each other, right? So you will have a long array of one type of residue at different positions. It's really hard to say where things go on the amyloid surface. And of course, the exact activity, if we see that amyloids have catalytic activity, maybe the levels and magnitudes of that and exact activities will vary with the fold of the amyloid that you study. so the polymorph will matter. Still, I showed you 2 model substrates and ATP. That's maybe you start to think about, what about biological substrates? So we decided to address that and see if we could see any activity on real substrates, metabolites from a cell. So here we took an unbiased approach to figure this out. We took neuronal cell lysate, sushi cells, and we took the lysate, we removed all the proteins, and then we just had these metabolites, the smaller things left, and then we incubated that with amyloids. And the idea was to analyze the metabolites before and after amyloid treatment, incubation for a number of hours. So to do this, we took a metabolomics approach, LC-MS, to identify metabolites. We did several biological replicas, we did different type points, we did different controls, monomers, amyloid, just buffer. So we had all these combinations. And then we looked what happened. So what we found in these samples were we could detect 2,000 MS peaks, so 2,000 metabolites with a different mass. Of those, we could assign 63 to specific metabolites that we could identify. These are the metabolites that we identified, and you can see it's a bunch of different things that are involved in metabolite reactions in cells that are, of importance, that has to happen all the time. So first we analyzed this data globally. We did principal component analysis of all the different samples. And here I show you the different samples, the PCA analysis. It's the same data, but I just color-coded it differently depending on what I want to emphasize. So if we just look at the plot to the right here, I circled the buffer and monomer samples at all time points. You see they all cluster there down at the bottom. But here, if we look at the amyloid-treated samples at 6 hours, the longest time point, we had 0, 1, and 2, 1 and 6 hours. You see that they shift away. So something happens in that amyloid-treated sample. You can also see from the middle plot here that we had 3 biological replicas, and the 3 different batches, they vary. So there's a shift there of all the data. So therefore, we normalized each metabolite to the time-0 value so we could compare the different batches. Then we did a t-test to determine for what metabolites of those 63 do we have a significant difference between monomer and amyloid treated samples. So we find that we have unique changes with 21 of those 63 metabolites. Here are the metabolites again, just a little bit bigger, so you maybe can read it. So the red ones here, these are metabolites that decrease in the presence of amyloid. There's 17 metabolites. The green are the metabolites that increase in the presence of amyloid. And I should say that in all of these 21 metabolites, the monomer treated samples, there was no change in the level of the metabolite. And for the remaining 42 metabolites, there was no change either in monomers or amyloid treated samples. So we only have a variation when we had the presence of amyloids. So I'll show you what went up and down. So these are the 4 metabolites that increased in presence of amyloids. We have NID, adenosine, and 2 different acids there you can see. And you see they go up at 6 hours and to different magnitudes depending on which one I look at. So the question now becomes, how are these made? Are they put together from smaller building blocks in the presence of amyloids? Or are they degradation products of a larger molecule that because of the amyloids in the sample, you create these, you remove something and create NID or adenosine and so on? We don't know, but it makes most sense probably that you have something bigger that is cleaved into these products. And based on the model substrate data, you could imagine that you can make NID by phosphatase activity on an NIDP, that would create NID. You can make adenosine by dephosphorylation of ATP if you remove all the phosphates, or also from hydrolysis of adenosyl homocysteine, which is a an important metabolite in our cells. When you think about these acids that are made, we could think about esterase activity on some kind of precursor that would result in an acid. Here are the metabolites that decreased in presence of amyloids. And you can see there are many that decrease, and we have several amino acids, aromatic amino acids, tyrosine, tryptophan, phenylalanine, and so on. And amino acid degradation plays a key role in the TCA cycle, in energy metabolism, in glycolysis. If you start to mess with amino acids, that can be problematic for the cell. And also dopamine, which is a signal substance that is made in the neuronal cells which die in Parkinson's disease. Dopamine is made from phenylalanine and tyrosine as precursors on the path to dopamine. So if we mess with those amino acids, that could be linked maybe. And then we also have, in addition to amino acids, you have acids involved in the TCA Krebs cycle. You have some other compounds here that I don't necessarily know exactly where they're involved, but you can find them in all these metabolic maps of metabolism of the cell. So the question here becomes, what are these amino acids and acids converted to? We see that they go down, but we don't know what becomes of them. And you can think about different types of chemistries involved. Maybe there's decarboxylation that could happen to all of these, or deamination, or oxidation. We don't really know. We're trying some things in the lab. We try to look for decarboxylation, but it's actually really hard to test this in an in vitro assay. I also want to point out that some of these decreases may be due to amyloid binding. We can't exclude that yet, because I have to spin down the amyloids in order to analyze the sample. So if something really binds to the amyloids, it will go down as well. But it's kind of hard to believe that all these 17 different metabolites will actually bind to the amyloids and go down. I don't think that's the explanation. So really, this is just the beginning. It's kind of opening a can of worms. There's so much more to look at. And remember what I said earlier, that we found 2,000 peaks, and I could identify 63 metabolites. That means that there are 1,900 something peaks, metabolites, that we haven't identified. And when we looked at the data for them, even if we don't know what they are, we find around 20 masses that go up or go down exclusively in the amyloid samples, that we now want to figure out how we can identify what that is. Because I think this is important to look into further. There could be biological relevance of these reactivities that we see. There are several metabolomic studies on Parkinson's disease patients, on experimental animals that mimic Parkinson's disease, on cell cultures that show that you have metabolome alterations in presence of synuclein amyloids in a more living system. And all these things are affected that I listed here. And many of these things that has been reported overlap with our pure amyloid incubation data. We just put in some amyloid and wait for 6 hours, and we see the same thing as somebody can find in a Parkinson's disease patient or in an animal. These things that I circled are at least things that are the same. I want to point out that lipids are also important to look at. We didn't look at lipids in our metabolomic analysis because you have to do targeted lipidomics in principle. But I think that's really important to look into. But maybe some metabolite changes in patients, in Parkinson's disease patients, are due to amyloid-mediated catalysis. So these amyloids that are formed during Parkinson's disease and other amyloid-related diseases, actually, they contribute to changes in the metabolites in those cells that can add on to disease progression. So the question is really, can this contribute to disease progression in Parkinson's, but also in other amyloid-related diseases? And does this mean that maybe catalytic activity is a general property of all amyloids, more or less? With specific activity activities depending on the fold of the amyloid, the surface of the amyloid, and of course the activity might be rather weak compared to real enzymes. But still, if you think about amyloid sitting in a brain for years, right, maybe a weak activity is active enough to cause some trouble. So before I stop, I just want to show you one more thing. So I mentioned before that we had looked at synuclein binding to DNA, and that we could see that synuclein monomers can bind to DNA and actually stretch the DNA. So then we speculated that it could be involved in gene regulation, something. But we now also found that alpha-synuclein amyloids can bind to DNA. This is, for example, Biocore data. It can actually bind to RNA as well, even if alpha-synuclein is not supposed to be an RNA binding protein. But the amyloids interact with DNA. And when they do, we looked at what happened to the DNA. And it turns out the DNA is damaged in the presence of amyloids. You can see that from this graph here. Here I use a repair enzyme cocktail in order to detect damages in the DNA, and that's a way to indirectly say that I have damages. So in presence of amyloids, the DNA is damaged, but not in the presence of monomers. So when we did this and tried to look, so this was an enzyme cocktail, so I didn't know what repair enzyme, it was a bunch of things. So if I separate these repair enzymes, we try to look at it in more details. And what we can see here is that there are two repair enzymes that become active when we have treated the DNA with amyloids or incubated the DNA with amyloids. That's endo4 and AP1. And these two enzymes detect the basic sites and nicks and oxidation. So something is happening here. I don't know if this is catalytic activity yet, because it's very hard to derive real numbers from these values that we get. So, they could be one turnover, more than one turnover. What's the mechanism? I don't know that either, but we think preliminary data suggests that reactive oxygen species are involved. So maybe DNA amyloid interactions somehow stimulate reactive oxygen species that damage the DNA. We have to do more here. But with that, I want to come to my last slide and show you my research group, my wonderful people that do all the work. Here they are. Ranjit Kumar, he's the research engineer that makes all the proteins, so we have this top-notch synuclein to work with. We also have Nikita and Istvan that works on catalytic activity, so most of the things that I showed you. Pinar, she's working on the fish protein that might be a putative cure eventually on amyloid formation. And then we have Gulshan, who is both a postdoc and works on copper interactions with the amyloids. And I want to point out Kadra because she's a former master student that started the metabolomics work. We have funding agencies, and I have Genie, a gender initiative that took a lot of my time for 4 years, which I should thank because they gave me some money too. And thank you all for your attention. Thank you.
Thank you, Pernilla. So we have some initial questions. Maybe I'll start with them. Or Margie, were you about to do that?
Well, yes. Thank you very much, Pernilla. That was super interesting and great to see some questions coming in. I thought I would just start with one of my own, if I could. So you mentioned that you were very careful about how you make the amyloids, the alpha-synuclein fibrils in vitro. And I wonder if you could just elaborate on that a little bit more, why you think it's important to be so careful. And also, second part, have you seen a difference if in the activity, in this catalytic activity, if you use sort of early stage fibrils and maybe some oligomers versus more mature fibrils?
That's a very good question. So first thing, I think we haven't varied. I mean, I don't know, because truly, I know these amyloid structures that are out there, right? And I can mimic the conditions. I have the right pH and the right buffer. And I think I have the structure that's similar to what somebody published, but I have never solved the structure of my own amyloids yet. So we do aggregation with the glass bead and shaking, then you get kind of a mixture of amyloids, and then we take that as a seed at quiescent conditions to try to get more. And from AFM, we can see we have more homogeneous amyloids. We could, this would be interesting to try to do it at a different, a different pH, a different buffer, and see by AFM how different the amyloids look like. And then does that relate to catalytic activity? It's a little bit, it comes back to the variants that I used here, because again, I don't know if these mutants, what are their structure in the amyloid fold, right? I make a truncation, I make a mutant that could affect the amyloid fold. So I think there's a lot of this structure function relationship that we need to go in and address that I don't know.
So that leads into one of the first questions, which is, why did you choose to study the H50A mutant?
Because synuclein has only one histidine, and early on we thought that maybe metals are involved. Maybe there are metals catalyzed. So then histidine will, be involved in copper binding, for example. So we also use that mutant actually for metal interaction studies. So that's why we had it, or we could try it. But then we also tried to do this catalytic activity, esterase and phosphatase activity. We added different metals, zinc, copper, iron, but it doesn't help. So it's not metal mediated.
Yeah, and potentially much more general than specific mutation.
And I think, I don't know if we were lucky, but I thought it was really amazing that we could see that the phosphatase activity was actually going down when we had this mutation. And we tried to do this ATP assay also. It's really hard to make ATPase just taking ATP. You can do this HPLC method that I showed you, but it's very hard to make time points. But we tried to do a spectroscopic assay. It's very unreliable, I would say, but it's starting to look now like this mutant, the histidine mutant, is bad also in, not just a model substrate for dephosphorylation, but also with ATP. So I think there is something to that histidine and phosphate.
So the second question from Shugung Zhang: can amyloid also carry out condensation reactions, namely chemical synthesis, for example, making a peptide from amino acids? And I think this is something that follows on from some early work sort of on the prebiotic soup and whether or not amyloids might form there.
So yes, and I think this is a really great question. And I think it might be possible. What we have tried is that we just took this, if you take the esterase activity and you make this yellow compound, and then you have, the other piece of it, and you just take the two pieces and you see if you can get rid of the color. You can reverse that, but we tried that and nothing happens. Of course, that can depend on conditions. Maybe we need to bump up concentrations, but we haven't seen any synthesis. And I think that's also going to be harder depending on how you bind on the surface, right? I'm thinking that peptide synthesis, for example, because amino acids, if you can bind the same thing just along a repetitive motif on the surface of the amyloid, then you can allow formation of bonds. But if you need two different pieces, maybe it's harder to put them together. But we have a lot of experiments to do here. So the question is always like, how can you do this in the test tube and have ways to probe what you get in a preferably easy way? Because many times you get back to HPLC and you need to separate things and figure out what happened. And you could, but then the problem is you need to get rid of your amyloids, so you need to spin down. So technically we have to solve a lot of problems, but it's possible.
Yeah, no, you made the point that it's difficult to be sure that you're not removing some exciting compounds when you separate your fibrils. Obviously, you can't inject them into the HPLC. So the next question from Patricia Soto: how generalizable is the conceptual framework to extrapolate from alpha-syn amyloids to others such as prion protein amyloids?
Well, I think we can generalize. We can think about it that all amyloids can do some kind of catalysis, then a little bit dependent on who they are. but I mean amyloid beta amyloids, glucagon amyloids, synuclein amyloids. I've heard about unpublished results for bacterial amyloids. And so, I mean, I think this is a general property. But I would love for people out there to go and take my simple model substrate and test it on your amyloids. We want to have all that data, so please do that and tell me.
So one of the things I've been interested in for a while, and which might cross over here, is melanin. Obviously, you know, we know about PMEL fibrils catalyzing melanin synthesis, and obviously there's melanin associated with these particular dopaminergic neurons that die in Parkinson's. I've been interested with fungal amyloids because there's a different form of melanin in the spores of fungal fibrils, and we've played around a little bit and had a suggestion that the fibrils at least were a platform for synthesis. I wondered if you had looked at that in your system.
I haven't looked at it, but I think I thought about it, I read about it, and maybe it can, you know, maybe the amyloid could actually do more than just be a scaffold there, or like, you know, so I think one should look at that and really do this experiment.
Okay.
Thank you.
Maybe so much. This is really exciting discussion. One of the things I love about these is the way we can have this discussion. So I hope we'll have more discussion. Maybe we'll go to our next speaker. Thank you, Pernilla.
So the next speaker will be Gazit, who will speak about metabolites and protein amyloids from newborn metabolic disorders to age-related neurodegeneration. So I think now we move further to like the smallest piece of an amyloid, maybe. Another twist on the amyloids that I were talking about, of the metabolites I were talking about.
Thank you so much, Pernilla. First of all, for organizing it and setting the stage also in terms of metabolomics as well as amyloids in general. I thank Elizabeth and the Protein Society for this venue. It's really wonderful. And so far, I also enjoy the discussion very much. So today I want to speak about metabolites and protein amyloids. I will start. Oops, my screen is stuck. Oh, for some reason. Oh, okay. You see the next one? Yeah. So more than 2 decades ago, we studied the simplest peptides that can form amyloids. It's a paper in Science in 2003, in which we recognized the diphenylalanine, which is the core recognition motif within the beta amyloid that already mentioned by Pernilla, that forms amyloid in the case of Alzheimer's disease, of all the molecular information needed to form ordered structures that look and behave like typical amyloids. And over the years, we study extensively the properties, how to inhibit those structures. And today, and this is our reductionist amyloid approach, starting from large 42 amino acid polypeptide, going to a hexapeptide, and then a dipeptide, as I mentioned. Still, tiny peptides that can form amyloids are full of surprises. This is from 2 years ago, when we finally solved the crystal structure of the diphenylalanine dipeptide, and we could see that even such simple dipeptide form typical amyloids with a canonical beta sheet like a formation, although there's only one peptide bond. Yet when we use inhibitors, it changes from the regular beta sheet-like structure into helical one, although the inhibitor itself is not in presence in the crystal structure, which provides us some information about inhibition, the ability to change the packing of building blocks that form amyloid structures. Another thing from last few years is our ability to identify short peptides that can go through liquid-liquid phase separation and form amyloids or precursor to amyloids. And once again, we could see the role of simple peptides, aromatic peptides, especially containing phenylalanine, tyrosine, and tryptophan, and so I could speak a lot about amyloids that are formed by tiny peptides, dipeptides, tripeptides, but we asked ourselves for many years, what is the minimal requirement for the formation of amyloids? And we thought about dipeptide, and the reason is the occurrence of the peptide bond, which could be in resonance and have some two-dimensional properties. And then something that really started as a control made a huge difference for us in the last decade. And as you will see, it's involving, I will combine both published work as well as unpublished work. So we tested diphenylalanine, we tested triphenylalanine, tetraphenylalanine, phenylalanine glycine, and others. And we asked ourselves whether a single amino acid, phenylalanine, could form ordered fibers. And the answer, of course, should have been no. But quite to our surprise, at relatively high concentration of the millimolar range, which are completely non-physiological, well, you will see a twist in the story, we could see things that looks like amyloid. They bind THT like amyloid. They bind Congo red, the indicative dye of amyloid, and show typical green apple birefringence. But all of this was done in concentration of phenylalanine of a millimolar concentration. The concentration of phenylalanine for most individuals is somewhere between 50, 60, maybe if you are a bodybuilder and you drink a lot of protein drinks, it's 80 micromolar. We're speaking about 1,000 micromolars. But then we realize that there are some conditions in which individuals may have millimolar concentrations of phenylalanine. This is in phenylketonuria. It's a genetic disorder in which individuals lack the enzyme that converts phenylalanine into tyrosine. And this results in a really high concentration of phenylalanine in blood and tissues. Actually, the clinical definition for PKU is one of these diseases that is tested for any newborn, is 1,200 micromolar of phenylalanine. And if this disease is not treated by special diet, you see all kinds of neurological, mental abnormalities. And as I said, the treatment is diet, which is really restricted with phenylalanine. And then we ask ourselves whether the phenylalanine could actually form toxic species, like we see with protein amyloids. And indeed, if we go to concentration in the millimolar range, we could get the formation of toxic structures, as we could see by cell toxicity assays. And we had a very good control in which we used alanine and not phenylalanine, which does not form structures. And this, you can put a very high concentration of alanine on cells, and you don't see toxicity, which goes against the common wisdom that was before a study that really the reason for the toxicity that is being observed in phenylketonuria is the fact that you have very large amount of amino acids. They block the transporters. They interfere with the balance of amino acid. But this is not in the case. Even if you put 15-fold higher concentration of alanine. Moreover, we were able to generate antibodies toward phenylalanine assemblies just by injecting those amyloids into rabbits, and we could deplete the toxicity by pulling down these structures using these antibodies that were made in rabbits. Moreover, we could go to brain banks, a brain bank of individuals with PKU, with phenylketonuria, post-mortem, of course, and we could see that they have deposits in the brain, looks like what you see in Alzheimer's disease, but that could be stained with Congo red, the indicative dye of amyloid, as well as the antibodies that we raised in the rabbits. So this was quite a surprising twist to the story of amyloid formation by very short elements, which was the first demonstration of a non-protein entity or non-peptide entity that can form amyloid-like structures. But then we scratch our heads and thought, well, phenylalanine, phenylketonuria is one example. However, unfortunately, there are many inborn error of metabolism disorders. These disorders in which individuals are defective in one or more enzymes in biocatalytic pathways. So we went very systematically, studying tens of different metabolites and tried to see whether they can form structures, especially amyloid-like structures. And indeed, we could see that not only phenylalanine, but also adenine, orotic acid, cysteine, tyrosine, uracil can form things that look like amyloid, bind THT like amyloid, bind Congo red like amyloids, and it appears to be that phenylalanine is only one example of many others. This is all unpublished results on something that was even higher surprise. This is a case of non-ketonic ketotic hyperglycinemia, in which individuals are accumulating glycine, simplest amino acid you can get, although I don't have the time to go into it. The packing of glycine is quite interesting. There are several polymers that can form. It was known before our work on amyloids, but generally the way that glycine could self-assemble. And this disease is very severe. It's affecting the brain, and it's a very severe neurological disorder. And also in this case, we could see things that behave like amyloid. I don't have the time to go into it, but we could see the formation of oligomers. This is another thing that we now beginning to unfold with mass spectrometry on the formation of these structures. So, the work that was done tens of years on protein and peptide amyloids, we now start to unfold on metabolite amyloids. And one of the interesting things is one of the criticism or questions that we get many times is, well, you see the formation of the amyloid in the test tube, but does it really reflect the situation in the patient? And in this case, we had a patient with glycine accumulation, and we had siblings, so with very similar genetic background, but they do not have the disease. And then we took sera from with the patients and the siblings, and we could see that patients have antibodies in their blood that recognize the glycine assemblies that we make in vitro. Also, of course, the rabbit antibodies recognize these structures, but these structures are not recognized by the siblings who don't have the disease. So it appears to be that the patients are exposed to these glycine assemblies, which means that it's really pathologically relevant structure, and it's very specific because their antibodies do not recognize phenylalanine, but recognize only the glycine structures. So this appears to be just as in the middle of the toe, slightly before the middle. We can form amyloid structures that look like amyloid by electron microscopy, bind indicative dyes, have many other properties that look like amyloids. They are toxic. We can raise antibody. We can see deposits in the brains of patients, and the patients develop antibodies. That means that they are being exposed to these structures. So in retrospect, in the beginning, people told us that metabolites are too small. How can they form things that are quite stable? But then we realized that in many diseases, amyloid and non-amyloid related, there are metabolites which forms deposits going from gout, kidney stones, gallstones, structures in the liver, so structures in the brain, as we mentioned, structure in the eye, like in hyperoxaluria. So it appears to be that metabolites could form structures, and people are now realizing, and once again, that this is quite a well-established phenomenon. Another unpublished work, so we thought, well, if we can get the formation of the structure by phenylalanine, and we see the correlation with toxicity when we see the deposit in the brain, can we inhibit the formation of these structures? So we launched a high-throughput screening campaign with 55,000 compounds. The readout was relatively easy. It's just showing that it could inhibit the formation of amyloid by by phenylalanine. We had few hundreds of hits, and then using pharmacophore models and in silico modeling, we came up with some lead compound to be tested with animals. So how you test if a compound is active? We have PKU model mice. Mice and humans have exactly the same metabolism of amino acids. So we have mice in which they are defective in the enzyme that converts phenylalanine to tyrosine, and they show cognitive and neurological deficiencies if they are not on special diet. And then we treat them with our compounds and try to see if we see some improvement in cognitive abilities. So one of the methods that is highly used for study of various neurodegenerative models is Morris water maze. You check the time that it get the mice to find the way in to find a hidden platform in a water maze, and we could see that by treatment we could shorten the time that it takes them to learn how to find a hidden platform. Another well-used test to test memory is Y-maze, because mice are quite curious. They want to go to new to explore places that they haven't been to. So it's a ratio between their exploration of a new place or as compared to old one if they don't remember. So testing with vehicle in a Y maze, they almost there's no preference to go into the new way, but if we treat it with a compound, we see a significant improvement in the cognitive abilities. All of this with having in the treated animals very, very high concentration in the thousands of micromolar concentration of phenylalanine. So the common practice to treat PKU, phenylketonuria, is to lower significantly to only few hundreds of micromolar the concentration of phenylalanine. Here we could see that even in the presence of high concentration of phenylalanine, if we have good inhibitors that are specific, effective in a nanomolar range, we could tolerate it, at least the mice could tolerate a high concentration of phenylalanine while significantly improving their cognitive ability as well as motoric activity. I didn't have the time to go into it. So other models that we have for amyloid formation are yeast models, which I will go into it towards the end of my talk, in which we could see genetic influence on the formation of structures, and it's as many of you know, yeast models are very useful to study all kinds of physiological processes. And we see here, similar to humans, the sensitivity of yeast to the feeding with amyloid in a very steep, if you look at D, in a very steep way. They could grow quite nicely when they accumulate, in this case, adenine. Also, we have adenosine related to what Pernilla mentioned. And when they reach a certain threshold, immediately it affects their growth, which is consistent also. Such type of sensitivity to addition of compound is usually when you have some kind of assembly process like you have with amyloids or amyloid-like structures. Now, Pernilla set the stage for metabolomics. I will go quickly. All kinds of methods are used to study all kinds of diseases and see the accumulation of various metabolites. So metabolites do accumulate in those inborn nerve metabolism into very large concentration. But also, as mentioned by Pernilla, it's in some of the neurodegenerative diseases, like those not newborn diseases, but diseases for old age, you also see abnormal concentration of metabolites. And then we ask ourselves whether these metabolites or metabolites amyloid could serve as seeds to make protein structures. So maybe seeding of proteins into amyloid structures by metabolites could clarify some unexplained epidemiological associations. And of course, all of you know that human metabolism is so complex. So many metabolites could be elevated or be lowered in different conditions. And this is actually even people with identical genetics could have different metabolomic signature. And one of the metabolites that is found to be elevated in Parkinson's disease is quinolonic acid. Again, it's in the tryptophan aromatic pathway. And we set to see whether quinolonic acid could form amyloid-like structures. And indeed, you could see that this looks like typical amyloids. And when we solve the crystal structure, we see the organization of like a beta sheet type of arrangement. And most importantly, when we check the interaction of these seeds with alpha-synuclein, alpha-synuclein, those of you who are working on Parkinson's disease, it's amyloid forming structures, but you need to little bit provide some mechanical agitation or force in order to have it. And with the seeds, we could see immediately the formation of amyloid by alpha-synuclein. So it provides at least some initial indications that there might be a crosstalk between the metabolite seeds and the proteins. Another thing related to Alzheimer's disease is homocysteine. Homocysteine is known to be elevated in the case of Alzheimer's disease. Once again, we could see the formation of things that look like amyloids by homocysteine. They have this here by crystallography a bit more complex structures in like a bi-layer type of a beta sheet, like two sheets that are on top of the other. And once again, here in mice models of Alzheimer's disease, the 5X FAD, which is quite a severe form of Alzheimer's disease, we could see the colocalization of beta amyloid, but we started with the peptides that form amyloids in the case of Alzheimer's disease and homocysteine. So once again, we have some indications about the crosstalk between metabolites and amyloids, and this is something that we continue to explore. So we see the severe case of the lower one, in which you have completely in dysfunctional metabolism. In early age, you could get the formation of structures and disease. And we think that in partial metabolism, some polymorphism, enzymes that do not act as good as they should, maybe you have some statistical chance to get the formation of the structure in advanced age. And this is how it's related to disease. I see that I'm off the time, so I would just mention quickly that we use the yeast in order to get information about mutation or overexpression of genes that can rescue the yeast cells from the activity of amyloids, and try to understand and trying to see all kinds of pathways that may be related to this, and this is again not published, but hopefully will be published soon. And I will just end up again just mentioning passing, because of Pernilla's beautiful work on catalytic amyloid. We were also very much influenced by the work on amyloid catalysis, and said, well, if we can form amyloid-like structures by these metabolites, amino acids and other metabolites, can we see some activity? And indeed, we could see some esterase activity, and later on other activities with also amyloids that are made of metabolites. Already when we spoke about dipeptides, I used to quote Leonardo da Vinci that simplicity is the ultimate sophistication. I think that when we went down into metabolites, this is really the case. So thank you for your attention, and I moved fast. So if someone is interested in the original work, he or she could look at our website. Thank you.
Thank you so much. This is so interesting that all these tiny metabolites can form amyloids, right? It's amazing that we are alive, actually.
Human metabolism is such a complex machinery. If something even slightly is getting wrong, and we get, unfortunately, all these very long list of inborn of metabolism disorders.
So would you think that the kidney stone is amyloids packed together?
I'm not sure. So this is a bit more complex. In those cases, you can have crystals, and this is also many things that all of us are in the also in the protein amyloid field. We also always have this discussion between pathways that leads to crystals, pathways to amyloids, how to handle it. And now, as I said, I'm coming from the Holy Land, so like a crusader, trying to convince more people to study metabolite amyloids. That protein amyloids represents, I guess, millions of scientists hours or years in research. Here we're trying to convince more to join us, because I think that this is quite
new. So do you think, I mean, so you showed a lot of different things that could form amyloids, of these like smaller compounds. Do you see any, and some did not, adenine or alanine didn't form anything. So do you see anything like a required motif that you need to get an amyloid?
Excellent question. So we could really see that the at least the minimal requirement, I don't know if this is the only requirement, but the minimal requirement in order to get the formation of amyloid-like structure is in crystallography, the ability to form layered type of arrangements, similar or equivalent in a way to beta sheet, like a supramolecular beta sheet. Those structures that form things that are in a different way, not a layered type of arrangement. There are, of course, other arrangements that small molecules could get. We don't see the formation of the structures yet. I don't know if it's, I know that it's the minimal requirement, but I don't know if it's the only requirement. And again, this is where we hope that things like now AI and studying large pools of metabolites could help us. We submitted a grant proposal on this. I hope that we could screen few thousand of metabolites and really try to understand the rules. But I have to confess that we only start to unfold this.
Yeah, I mean, it's new and you need more people out there to study this. And I'll read one question that came in here. Actually, people out there, add more questions because we have time for that. But this is a question: do tryptophan and guanine at high concentrations form amyloid? Tryptophan and guanine.
Okay. So we tried both. This goes well also to your question, earlier question, Pernilla.
Yeah.
This is always the competition between amyloids and crystals. So actually, guanine and tryptophan tend more to form crystal-like arrangements rather than amyloids, probably because the aggregation is too fast and it reaches this minimal energy. Guanine by itself, it's a very interesting. Shugwan, of course, knows it very well, but guanine by itself, it is being used physiologically. We are speaking. I cut more and more slides, but we will hear from later on about from Matt about functional amyloid, and we also there are functional metabolite assemblies. Guanine is one of the examples. Physical colors, the chameleon's way of changing the color. So again, so these are more tends to form the some structures, crystalline like assemblies, and less the formation, less amyloid or pathological amyloids.
So I have a practical question. I mean, if I'm thinking that I should, help you and try to do some of these metabolite amyloid experiments, you need really high concentrations, right, in order to make the amyloids. But can I then take that amyloid, and if I want to make a seed and put it in solutrin or AB or something else, can I dilute it and use it at a lower concentration, or does it dissolve?
Well, it is not as stable as protein amyloids, but to some extent you can have it. It's yes, you can make it high concentration and then dilute it. Yeah.
I was going to ask a similar question because you were able to inject the fibrils into rabbits and raise antibodies, so they're stable enough, presumably for antibodies to develop, and that's really interesting too. Do the patients have autoantibodies against these metabolite amyloids?
Okay, maybe I moved too fast. Yes, they do have. This is unpublished results. We are extremely excited about this. We see the formation of antibodies in the patients, and as I said, maybe too quick, we have a patients and siblings who do not have the disease, just the same genetic background. They recognize glycine assemblies, the patients. The siblings do not recognize, don't have antibodies to recognize it. and both of them don't recognize phenylalanine assemblies. So it's specific antibodies toward glycine. So we are very excited. It's now the manuscript is under consideration.
Yeah. Sorry, yeah. I do recall that you said that. It was raising a question for me. You know, I'm going to talk about some mammalian functional amyloids that we don't raise antibodies to. So it's really striking, isn't it, that that these,
because if we make amyloids antibodies to it, right, it means something. If you find it in patients, it has to be there. It has to be a point.
So from our point of view, it's the patients are exposed to these structures. So it's not only in the test tube.
But okay, maybe a final question then, because maybe we should move on after this. But I mean, so you form these amyloids in these diseases, but at the same time, you have lack of something else, right? For example, phenylalanine should become tyrosine, right? But you don't make any tyrosine.
Yeah.
So you both have loss of something, and then you have these amyloids or too much of something. Yes, so can you separate like all these problems that patients have in, like, what is most dangerous or?
So in under normal nutrition, they just get the tyrosine from. That's
not the big problem. I mean that's clear. The biggest problem is building up of
because they have it in the diet, and if they are on a special formula, once again they get the tyrosine from the formula.
Okay, so it's really, I mean, then you come back to it must be the amyloids that are causing trouble.
At least our indications. I have to be careful. Yes.
Thank you so much, Udi. I think we will move on to the next speaker because I can see our time is going on, but we don't have to stop exactly after 2 hours, so we can go on longer. And we will finish this, you know, with last. The next speaker is Margie Sunde. She will speak about functional amyloids on either side of the host microbe divide. So now we'll go into microbes, bacteria, maybe. So go ahead.
Thanks very much, Pernilla, certainly for organizing this great webinar, and to the Protein Society as well. So, yeah, switching a little bit, I'd like to tell you about some of our work on functional amyloids produced both by fungi, viruses on the sort of microbial side of the battleground, and mammalian amyloids on the other side. And I'd like to start by paying my respect to the elders and communities past and present of the lands that the University of Sydney campuses stand on, and that's the Gadi people of the Eora Nation. So I'm going to tell you two stories today. First of all, focusing, excuse me, on functional amyloids produced by filamentous fungi. And I might call these rodlets interchangeably with fibrils, because mycologists, as you can see in this image on the left-hand side, mycologists have known for more than 50 years that the spores produced by fungi are coated in a tightly packed fibrillar rodlet layer. But there was not much further investigation of them until the early 2000s, when people recognized that actually these protein fibrils had an amyloid structure. And what's a little unusual about these is that they pack together very tightly to form layers, which is distinct from disease-associated amyloids and perhaps other functional amyloids. And also these layers are amphipathic, and that's part of their function. So on the outer surface of the spore, they're really hydrophobic, and on the inner surface of this layer, in contact with the cell wall, they're hydrophilic. And then the second story that I'll tell you about is some other sorts of functional fibrils, which I've chosen because they're unusual in that they're heteromeric fibrils. So whereas most disease-associated fibrils, and even these hydrophobin rodlet ones, are composed of the same type of protein, so multiple copies of the same type of protein. These rim-containing fibrils, which are found both in mammalian systems, but also, as I'll show you, produced by viruses, the fibrils that they form are heteromeric. So we're used to proteins having single amyloidogenic motifs, which are held in check in some way in the folded form of the protein, or if the folded protein's intrinsically disordered, they don't assemble spontaneously, but they do when they, under amyloidogenic conditions, the amyloidogenic motif, which I've shown here in green, forms the core, the beta sheet core of the proteins. In these rim-containing fibrils, different proteins share similar and closely related amyloidogenic motifs, usually in conjunction with large folded globular domains. And under certain conditions, these different proteins can come together and form an amyloid core. But here the amyloid core is made up of that motif from different proteins. And this was first reported in a mammalian system in this signaling complex known as the necrosome by Hao Wu's group in 2012. But switching to these functional fibrils on the surface of fungal spores, filamentous fungi tend to grow in moist environments, and they need to move out of the moist environment into the air in order to produce spores when they're reproducing asexually. And so hyphae grow through the air-water boundary and then produce conidial chains or spore chains. And this is where these hydrophobin proteins come into play. On the one hand, hydrophobin monomers can be secreted into the aqueous environment, and these small hydrophobin proteins are themselves amphipathic, and they associate at the interface and act like a surfactant and reduce surface tension, allowing the aerial hyphae to grow up into the air. And then hydrophobins are produced again when the spores are forming, and they produce these coats on the surface of the spores. And if you look on the right, you can see these protein layers coating each of the spores. And as I said, this protein coating is responsible for the fact that these spores can remain dry. And the way we know this is that work done by Beaver et al. In New Zealand in the 70s, they were able to knock out the gene for this small protein, this small hydrophobin protein. And what they discovered when they knocked this gene out was that instead of the hyphae being highly water resistant, so here these are water droplets sitting on hyphae and they bead up because the hyphae are so hydrophobic. When you knock out this gene, now the water droplets soak in very readily because the surface of the hyphae and the spores lack this hydrophobic protein layer. So it seems that the purpose of this polymeric amyloid layer on the surface of the spores is to make them water resistant. And this allows them to be dispersed readily in air over long distances. And we're all familiar, obviously, with the way that fungal spores can spread, leading to infection over long distances. I'll focus initially on the sort of water-resistant coating, but these amyloid fibrils, as we know, are highly proteolysis resistant. And this plays an important role in the rodlet amyloid layer biological activity on a human pathogenic fungus. But just stepping back to this protein that we chose to work on first, and this is work in collaboration with my colleague Anne Kwan, this protein, small hydrophobin protein, which was named EASE, which is short for easily wettable, which was the phenotype of the gene knockout. Turns out that EASE is a small soluble protein with a beta barrel held tightly by 4 disulfide bonds, has 2 long loops, it's very soluble and stable in solution as long as it never sees a hydrophilic interface. What's really striking about this molecule is that it has a very amphipathic surface. So one side of the protein contains all of the charged residues, whereas the other side of the protein is almost all hydrophobic. And this is really the key to this protein's behavior. As I said, in solution, when there's no hydrophilic interface, the protein is soluble. However, if you expose it to air, and we do this in the lab usually by shaking solutions or bubbling air through, then you can have the monomeric protein spontaneously assemble into these ordered amyloid fibrils or rodlets. And on the right is an electron micrograph showing the structure of these. And these bind thioflavin T and Congo red, as you would expect. So we can take that and use it in our assay. So what we're doing here is that we're maintaining the proteins in solution, and we're shaking the solution, but we've capped each well in the plate very tightly with a film so that there's no air-water interface. However, if we stop the assay and introduce a little bit of air, continue shaking, we get rapid assembly. And we can probe this, this assembly process, and we can see that during the process of assembly, the protein goes from a more right-handed twisted beta sheet structure into a relaxed anti-parallel beta sheet form. Ingrid McIndoe, working with me, carried out a mutational analysis and was able to identify the parts of the protein that are responsible for forming this amyloid. And it turns out to be in one of the loops of the protein. And our model for this is that the protein in solution doesn't undergo intermolecular assembly, but when it reaches a hydrophobic interface, such as the air-water interface, it undergoes a conformational change. You know, some proteins denature at a hydrophobic interface. In fact, this is a functional change for the hydrophobin, and this small amyloidogenic motif is exposed and allows it to line up with the similar, well, the corresponding motif in other molecules, and we get the growth of this beta sheet-rich structure. So now putting this all together, we can see how these hydrophobin molecules self-assemble into these long amyloid fibrils. And when we look in the layers, we can see this sort of double track structure, which shows the beta sheet-rich core and then the rest of the globular parts of the protein. And it's interactions between the globular parts of the protein that allow the proteins to stack laterally and make these layers. Oh, I should also show you that we can reiterate the amphipathic nature of these surfaces. So we know that the outer surface is hydrophobic, whereas the inner surface is hydrophilic. And in terms of biological function, this unique control of amyloid assembly is really important, and it allows rodlet formation to be limited to a hydrophobic-hydrophilic interface. And in functional amyloids, the organism doesn't want to produce these highly stable amyloid fibrils at the wrong place or in the, at the wrong time. And this is actually data from another hydrophobin, which is involved in infection of rice and other grains. But I'm just showing these data to highlight some of these interesting features. So as I've shown you before, we don't see hydrophobin assembly until we introduce an interface, a hydrophobic-hydrophilic interface, and this can be the air-water interface, or it can be hydrophobic surface, such as graphite. The rate of elongation is tuned to the area of the interface. So if we keep the solution volume the same, but in fact, we go to a 48 well plate where the area is larger, we can see that using thioflavin T assembly assays that the rate of assembly is faster. And if we shake the solution faster so that we're renewing that air-water interface more quickly, we see more rapid assembly. And what's really unusual for people who are used to seeing nucleation-dependent polymerization for amyloid fibrils, certainly disease-associated amyloids where there's a lag phase, here, the rate of elongation is completely independent of the hydrophobin monomer concentration. So what this shows is that it's the interface which is limiting in this reaction, not the concentration of the monomer in solution. And that's because the conversion to the amyloidogenic form strictly only happens at the interface. So now I'll show you how some of this is involved in a pathogenic environment, actually in the biological function of the hydrophobins. And this is the pathogenic fungus Aspergillus fumigatus. We've done this work in collaboration with Jean-Paul Latche, who was at the Pasteur and worked for many years on Aspergillus. So in 2009, Jean-Paul's group showed that the surface hydrophobin layer on Aspergillus fumigatus spores really prevents immune recognition of spores. So you and I would be breathing in several hundreds of these spores every day. And for healthy people, this isn't a problem. The spores are ingested and killed by circulating immune cells. However, for people who are immune compromised and lack circulating immune cells, the fungus can actually generate in the lung, forming aspergillomas or going on to invasive aspergillosis, where this disseminates into the blood and into the brain. And this is clearly a problem. So the reason we don't mount a continual immune response if we have a healthy immune system is the presence of this hydrophobin rodlet amyloid layer on the surface of spores. So on wild-type fumigatus spores, again, we see this tightly packed fibrillar layer. And in dormant cornidia, shown here, where the layer is intact, if you expose macrophages to these sorts of spores, you don't see any response measured as an induction of cytokines, here TNF-alpha. However, if you remove the polymeric layer and now expose the pathogen-associated molecular patterns in the cell wall, such as in this genetic knockout, now these spores do stimulate a strong immune response. The same is true if you remove the layer chemically. You need to treat these spores with hydrofluoric acid. The polymeric hydrophobin layer is extremely robust, but then these naked spores do stimulate the response. And germinating spores where the hydrophobin amyloid layer is lost also do stimulate an immune response. However, the protein itself is non-immunogenic. So we were really interested to understand both how the hydrophobin, in this case, it's a hydrophobin known as Rod A, which forms these polymer layers, whether or not we could understand how which parts were important in making the amyloid, and also how that played out in terms of biological function. And so Jen Lai carried out a series of immunogenesis experiments and was able to identify that in this case, both there are two loops in the protein, and they're both important, both in rodlet assembly and in the final structure on the surface of the spores, as I'll show you. So mutations in these two loops change the process of assembly from something really rapid in our in vitro assay to having a very long lag phase, because now these motifs are not as amyloidogenic. And Jean-Paul and his team were able to recapitulate these mutations in Aspergillus fumigatus strains and see the effect in production of the polymeric layer on the surface of the spores. So whereas the wild-type Rod A layer shows the existence of this tightly packed protein, protective protein coat after 3 days, this is not true of these two mutants. And although the single mutant shows after 30 days a fairly well-coated ordered layer, there's still disruptions in the double mutant, and this is reflected in these assays looking for cytokine activation in immune cells, where the wild type spore doesn't stimulate the immune system. But if you don't have a really intact and stable rodlet layer in these two mutants, you can see you get production of cytokine, things like TNF-alpha and interleukin-6. So just to summarize that, in terms of filamentous fungi, they're making amyloid fibrils that have very similar structural features to disease-associated fibrils. However, the assembly needs to be tightly controlled, and it's restricted to an interface where the fungus interacts either with the host or its environment, makes this protein barrier which protects spores from host immune recognition, and this allows the fungus to persist in certain niches, such as in an immune-compromised individual, and then germinate and invade. And I haven't shown you other data where we've shown that this protein layer on the surface of the spores actually facilitates adhesion of the spores and infection of the host. So just now, changing to that second really unusual system of functional amyloid fibrils to highlight some of the differences really between functional fibrils and other types of amyloid. I have to give you a brief introduction to programmed cell death by necroptosis. And this really is a cellular system which is employed by the organism for protection. So if individual cells are damaged through things like inflammation, which would trigger the death receptors such as the TNF receptor, bacterial or fungal infections, which would activate toll-like receptors, or undergo viral infection, then it can be advantageous to the host to switch on a pathway of organized programmed cell death and kill that individual infected cell. And what happens in this case is that these three adapter proteins recognize the danger or the damage to the cell. They become activated and go on to interact with this downstream kinase known as RIP kinase 3. I don't want to go into too many details about these proteins, but they all contain a small amyloidogenic motif known as a rim. I've highlighted this here with these black lines. And these rims are 18 residues long, and they have a highly conserved core tetrad. And how Wu's group was able to show in 2012 that this is actually a fibrillar amyloid complex. And what's unusual here is that, as I said, it's heteromeric. So we have copies of the adapter proteins as well as this key kinase RIPK3 present in these amyloid fibrils. And the globular domains remain active in this case on the outer surface of the fibril. RIP3 becomes phosphorylated and goes on to phosphorylate the pseudokinase MLKL, which then lyses the damaged cell. And how Wu and Miguel Mompian and Anne McDermott were able to show the structure of this amyloid core and this was published in 2018. But what we also know is that certain herpes viruses produce proteins which contain rim motifs, which are similar to the ones we see in mammalian proteins. And they are delivered into the infected cell when viruses infect, and they're able to inhibit this process of programmed cell death. So it's really a battle between the host cell recognizing the viral infection and trying to kill that individual cell to protect the whole organism, and the virus, which wants to prevent cell death long enough in order for the virus to replicate and disseminate new viral particles. So my group was really interested to try and understand how these viral proteins, which apparently have motifs which allow them to form amyloid, are able to inhibit another amyloid complex. And what we showed, to cut a lot of hard work by many people short, is that the viral protein, in this case focusing on this viral protein M45, is not a simple inhibitor. It forms amyloid fibrils in the same way as the mammalian proteins. But when it's in the presence of the mammalian proteins, it doesn't inhibit them from forming amyloid, but in fact, it sequesters them into large fibrillar networks. The same is not true when the two host proteins interact, making relatively small and defined amyloid fibrils rather than these large networks. And we really wanted to understand this further. So what we did was label our host and viral proteins with different fluorophores and then mix them together under conditions where we could measure, we could determine interactions between viral proteins and host proteins, in this case colored with a YPED tag or an M-cherry tag as green or magenta. And we used something called confocal coincidence spectroscopy to analyze the makeup of these different complexes. So to be able to determine were they homomeric, so just one type of protein, or heteromeric, carrying both partner proteins. And what we were able to show that indeed the viral protein specifically interacts with the host protein. And this is shown by having the magenta color and the green color here. So both fluorophores present in very large complexes. And if you mutate that core tetrad in the viral protein, you don't see any assembly with the host protein. And here what we've done is we've just excited the host protein. So we're just seeing what happens to the host, that key kinase, RIP kinase 3, when you add in the murine protein. In this case, we're not using the fluorophore on the murine protein. And what you can see, hopefully, is that all of the host proteins sequestered into large complexes, whereas if you add in the mutant protein, you don't impact the host protein. And indeed, using super resolution microscopy, we could show that the two proteins are in these fibrillar complexes. We then went on to try and understand how the viral protein can outcompete the other host proteins, which would normally be signaling for cell death. And indeed, we were able to show that the viral protein shows a preferential interaction with the host protein RIP3 over the interaction between RIP1 and RIP3. So in this case, the viral protein sequesters the host protein and actually displaces the RIP1 fibrils. It's not only murine cytomegalovirus, as I said, a number of different viruses encode proteins with RIMs, including varicella zoster, which is the virus which causes chickenpox. Won't go into too many details here, but in fact here the RIM is contained in a triplex protein, which is one of the key viral proteins which maintains the structure of the viral capsid. So when the virus infects the host, this RIM is delivered at the same time. It has a similar RIM, it forms amyloid fibrils. In this case, the viral RIM doesn't interact preferentially with RIP3, but in fact it interacts with that viral sensor ZBP1, forcing it into very large fibrils that are heteromeric, containing both the viral protein and ZBP1. And in this case, we've carried out a number of infection assays. And what's unusual here is that this system doesn't actually inhibit necroptosis, but inhibits another, perhaps more well-known form of programmed cell death, which is apoptosis. And this is probably because caspase 8 remains active in those systems. So just to wrap up, here the RIM fibril assembly is unusual, and that's it's heteromeric. It's really a functional system on the mammalian side of things, allowing it to maintain the health of the organism when under attack from inflammation or different forms of infection. But viral proteins actually preferentially interact with host proteins, and instead of the protein forming the RIP1 or forming a complex with RIP3, it sequesters them into alternative fibrils. We don't really yet understand how this programmed cell death pathway is controlled. There must be mechanisms by which these highly amyloidogenic motifs are maintained in a non-assembled form until the viral or bacterial or fungal insult is delivered. But that's work for the future. Anyway, I'd like to stop. I hope I acknowledge most of the people throughout the talk who did the individual experiments, but a very large number of people have contributed to the work, and many other collaborators have been involved. So thanks. I'd like to stop there and answer any questions.
Thank you so much, Margie. This is fantastic that, so actually we are alive or survive all the time against microbes because of amyloids.
Yes, and that's why I find, Ehud's amyloids so interesting, because, they are clearly recognized as deleterious and antibodies are raised against them. But we don't have circulating antibodies against these functional fibrils, which are active in our cells. To protect.
So how does the body know what's right and wrong, right? What do we want and not? And it's still our own pieces that we put together.
Yes, and I think our cells have mechanisms. Perhaps it's a rate of turnover. Perhaps it's because all of the fibrils remain in the cells and then are dealt with, perhaps by autophagy. Those are questions we're starting to understand now. Are they? I think that functional mammalian fibrils are probably less stable than the pathogenic ones, because they don't need to be any more stable than to last long enough to signal for the cell to die. So perhaps
you don't need more, right?
Yeah. We haven't yet identified any proteases that are specific, but there have been some reports that we may have chaperones which are specific for turnover of functional amyloids. Perhaps they become overwhelmed with disease amyloids, or perhaps they actually are sequence specific. And I think we're just at the start of trying to understand.
Could there be actually like chaperones that help these functional mammalian amyloids that we don't know of yet, that we could still? Sometimes I feel like we would know all the genes that are there, right? But could there still be unknown proteins there that maybe eventually we can exploit in diseases?
Yes, I think there might be, or we may not have yet exposed the chaperones we have identified to these sorts of substrates. So I think there's an awful lot of work to be done. And I'd be interested to know if, Ehud, if any of your metabolite amyloids are vulnerable to chaperones.
I have a lot to ask you. If I have time for one question, going back to the immunological response, is it possible that these functional amyloids have a role early on and they somehow been recognized as self, and there are some clonal selections to recognize them as self in a sense?
Yeah, that may be correct. There's a little bit of evidence in mice that damaged cells will switch on necroptosis, and so that might happen early on, and then be a way of training the immune system during sort of not quite homeostasis, but early on in development. There is a role for this.
Okay, so we actually have a question here from Shuguang again, which is great. More questions, we want that, right? Because we can answer them when somebody talks as well, or typing. So Shuguang asks, do most spore-forming microorganisms have similar amyloid proteins? How about mushroom spores? Do they also have similar spore amyloid proteins?
Yes, absolutely. Great question. So they do. And we're all, people who eat mushrooms are eating hydrophobin amyloid rodlets all of the time. Not only are they on the spores that are dispersed, but in the gills of mushrooms, hydrophobins provide a waterproof but gas permeable membrane, allowing gas transfer in mushrooms. So we know they're certainly not toxic when ingested, but really good, really great question. And there are also some bacterial spores that have similar types of polymeric amyloid structures.
Wow, I think this is just amazing. There's amyloids everywhere, right? It's a perfect field to go into for young scientists picking what to do in the future. I think in the interest of time, we should move on. We have a final speaker. The last but not the least, we have a final punch here from Matt Chapman. And you will speak about bacterial functional amyloids and link it back to neurodegeneration and human disease, likely. So go ahead, Matt.
So thanks, Pernilla, and thanks so much for organizing all of us to speak in this, and thanks to the Protein Society for giving us this forum. It's really been great to listen to everybody. So I'm going to maybe try and connect some dots between these functional amyloids and maybe their potential interactions with the traditional disease-associated amyloids that I think probably everybody is familiar with. So neurodegenerative diseases, I'm a microbiologist, so I put this slide up, made this slide mostly for myself to remind myself a little bit about neurodegenerative diseases. It includes dementias, and the most common of those dementias are Alzheimer's and Parkinson's disease. Just those diseases have a huge economic impact and affect many people, 55 million or so people worldwide. That number is only going to go up. These are age-related diseases. You can see down here in these two graphs that as we age, we become much more susceptible and much more likely to get both Alzheimer's and Parkinson's disease. And as our population ages, as we get better at treating other forms of infectious disease, it raises the likelihood that we could be susceptible to these age-related neurodegenerative diseases. 60 or over 50%, 60% or so dementia cases right now are in low and middle income countries. So this is despite the fact that these are somewhat harder to diagnose in some cases than other diseases, and this is only expected to rise by as the years go by. So this is a worldwide problem and a big problem. They're multi-causal. There's lots of ways I think you can presumably get to these dementias, but they're collectively referred to as protein misfolding diseases. And the reason for that is, you think of protein misfolding, we've been talking a little bit about it as we've gone through the other talks. A polypeptide, will start out in some sort of disordered form and hopefully fold into its native fold. And even if it misfolds along the way sometime in the cell, it can be chaperoned back onto pathway or degraded. And this is what happens most of the time. But every once in a while, a protein will adopt a really stable conformation. And the stable conformation that we've been talking about today is this amyloid fold. It's obviously a very conserved protein fold. It is present, lots of different proteins, as you've seen today, can get into the amyloid-like fold. And then it's a very stable fold. So once it gets there, if it gets into that fold, can sequester chaperone machinery inappropriately and really start to cause a cascade. And many of these diseases that you're familiar with are caused at some point, or at least a hallmark of those diseases is the formation of amyloid. And so where does the toxicity come from? And this is sort of a little bit of a charged question, because I don't think we exactly know in every case, but certainly there are some indications as to where the toxicity in amyloid-forming diseases comes from. So amyloids can be highly toxic when they form at the wrong time or the wrong place in the cell. We've seen very good examples of amyloids forming at the right time in the right place and cause no toxicity. But when you put them out of context, in cases if a protein misfolds and forms amyloid inappropriately, that can lead to toxicity. The folding intermediates tend to be the most highly toxic species, or at least in many cases. So if a protein starts out as an unfolded polypeptide and then eventually gets into a fiber, it's these intermediate states along the way to a fiber that can oftentimes be the most toxic species, and they can interrupt membranes and sequester cellular chaperones or make other inappropriate interactions with proteins or molecules in the cell. Not all amyloid is bad. Of course, we've heard lots about these functional amyloids where maybe the native fold of the protein is actually this amyloid conformation. And there are really hundreds and maybe even thousands of examples of these functional amyloids now. And to steal a term from David Eisenberg, Nature's amylome. I've had a slide that looked like this for the 20 or so years that I've worked on functional amyloids. And the first question here was completely rhetorical at one point, which was, are functional amyloids the rule or the exception? We had, of course, had many decades of research on the disease-associated amyloids like A-beta that causes, or at least is associated with Alzheimer's disease, and alpha-synuclein associated with Parkinson's disease. We had so much robust research on that. And these functional amyloids, as they were first being discovered, were clearly the exception. Although I think they are the rule. There are many more of these functional amyloids, I think, out there in our cells and running around in nature than there are disease-associated ones. And these span all aspects of cellular life, from bacteria to yeast and animals. I'm going to focus on these functional amyloids that we've studied in my lab for a long time, called curli, which are made up of this CSGA protein. But collectively, I think many of these functional amyloid systems hold promise for understanding how to control amyloid formation, how to control the timing and the placement, to put it at the right time in the right place in the cell, and the efficiency of amyloid formation, so that maybe you could skip over some of those toxic intermediates that can be associated with the disease states in amyloids. And so we've done that using these curli fibers. These curli are made by enteric bacteria. They're important for biofilm development. So here is a picture here of a biofilm that's forming at the air-liquid interface in a statically grown culture of E. coli bacteria. The E. coli are producing curli, and these curli allow the bacteria to connect to each other and then form this layer at the air-liquid interface. And then if you knock out the curli just genetically, the bacteria still grow fine. It looks like it's a blank slate there, but the bacteria are growing fine. They're just not making curli, not making the pellicle biofilm anymore. These curl-eye fibers help to protect the cells from other stresses. They're potent immune modulators. The major protein is the CSGA protein. The CSGA protein, interestingly enough, is secreted from the cell as a completely disordered or intrinsically disordered protein, which is quite unusual for bacteria to secrete an unfolded protein. And then the protein folds on the cell surface into this amyloid-like fold. And then these amyloid fibers, these curl-eye fibers, have all the properties of other amyloids. They're beta sheet rich, they're extremely stable, they form fibers that look like basically any other amyloid fiber that you would want to put on the slide. And so we can manipulate this CSGA protein quite readily in vitro, so we can purify this CSGA protein away from the cell. And when we purify it away from the cell, it starts out as this intrinsically disordered state. And then in just a couple of hours, it starts to form visible fibers by TEM. And then these mature into what we call the mature amyloid form of the curl-eye fibers. We can measure this amyloid formation using a thioflavin T assay, which is a standard assay in the field, where you can see sort of a lag phase of amyloid formation. If you look at the blue line down here, and then amyloid formation dilly-dallies around and then goes into a fast phase and then a plateau phase. And you can overcome the lag phase of amyloid formation in the test tube by adding some preformed curl-eye fiber seeds. And so if you add some preformed amyloid fibers, they can template the formation of the CSGA into a fibril. And this is going to be important later. This process of templating the formation of amyloid, it's called seeding. And it turns out that all amyloids, or most all amyloids, can be seeded. All the ones that have been looked at, I think, results in a rapidity of converting the monomer to a fiber. And seeding may contribute even in disease progression during neurodegeneration, because once fiber formation starts, it's a self-fulfilling prophecy that the new subunits will be accumulated into the amyloid-like form. And of course, the prion diseases are great examples of this seeding in action, because the prion diseases can be transmitted via a seeding-like process. Okay, so my lab has spent a long time and really dedicated ourselves to trying to understand how do functional amyloids polymerize at the correct time in the correct place. And so I think this is a pretty picture, in part because it makes a point. So you've got these bacteria that are producing thousands of copies of a highly aggregation-prone subunit, CSGA, or a protein called CSGA. And the bacteria have to secrete that amyloidogenic subunit through two membranes and then get it to the cell surface, and then at the cell surface have it polymerize into this beautiful amyloid fiber. And so how do they do that? And I'm going to skip over most of the work my lab has done so that I can tell you a kind of a quick story about the connection between these and eukaryotic amyloids. But when we look at the biogenesis, or what we know about the biogenesis of these curl-eye fibers, we've got the inner membrane of the bacteria here and the outer membrane here. And these proteins are made in the cytoplasm. And we've shown that there's some chaperones in the cytoplasm that make sure they get ferried correctly to the secretion machinery. That gets them into the periplasm. But then it's really interesting, in the periplasm, there's another suite of specific chaperones, CSGE and the CSGC protein, which are really important for keeping CSGA from forming amyloid inside the periplasm before it gets secreted to the cell surface out here. And these proteins are quite remarkable because they have to work without an ATP or energy source. And so they have to work just through protein-protein interactions with the subunits. And so we think these are really interesting anti-amyloid agents. And so, but then the CSGA does get secreted. Again, it gets threaded through this CSGG secretion machinery. as a unfolded polypeptide gets to the cell surface, and then it polymerizes into an amyloid on the cell surface. And so once these fibers are made, there are biophysical similarities between functional amyloids like curli and disease-associated amyloids like alpha-synuclein. We heard Pernilla talk about alpha-synuclein in her talk. And we've wondered for a long time, is there any biological connection between these different amyloids, human proteins that form amyloid and maybe these bacterial amyloids that could be part of the microbiome? And Parkinson's disease, which is caused by, or at least a hallmark of Parkinson's disease, is the amyloidogenic aggregation of alpha-synuclein. It's associated with severe motor dysfunction, this disease, but the non-motor symptoms precede motor symptoms by years and sometimes even decades. And approximately 80% of patients have some symptomatic gut dysbiosis. And this has certainly suggested to many people through the years that the early onset of gastrointestinal dysfunction in Parkinson's disease suggests that there may be a link between the bacteria in the microbiome, or what's going on in the microbiome anyway, or the gut, and what's going on eventually in the brain, because we see these effects really start in Parkinson's disease in the gut. And so there's been some really nice studies through the years to sort of link the brain and the gut in these diseases. So for one, alpha-synuclein fibril aggregates can migrate from the gut brainstem via the vagus nerve. They can migrate up, and so it's not the fibers themselves that are migrating up, but the propagation of the fibers, which is migrating up. So the fibers are seeded in the gut, and then new amyloid formation is able to add on to those to propagate those up to the brain. And Brock's hypothesis is that some unknown pathogen in the gut could be responsible for the initiation of sporadic Parkinson's disease. And then I think in really a seminal paper back in 2016, the Mazzanium group showed that the human gut microbiota from Parkinson's patients could induce enhanced motor dysfunction in a mouse model of Parkinson's disease. And this is where we kind of came into it, because we saw this and interacted with Sarkis. I'd known Sarkis for years, and so I knew this study was coming. And I asked him, could a bacterial functional amyloid like curli be a culprit through a seeding-like mechanism with alpha-synuclein? And so Tim Sampson in Sarkis's lab set up the following experiment, and I think it's a really, really brilliant sort of result here. So what he did was we gave him curliated bacteria or wild type bacteria, and then the same strain that was missing the curli gene, so it could no longer assemble curli fibers. And he put those two strains into either wild type mice or mice that were overexpressing the human form of the alpha-synuclein protein. And then he looked in these mice for different Parkinson's-related dysfunctions. These are mostly motor tests for the mice: time to cross a beam, and time to descend a beam, and time to kind of raise up on the hind legs and remove a sticky note from their nose. And as you can see, in the alpha-synuclein overexpressing mice that got the curli plus bacteria, in each of these cases, these mice are more deficient for those activities than the curli deletion strain and or the wild type mice that contains the curlated bacteria. And so curli producing gut bacteria enhance the pathophysiology of Parkinson's Parkinson's-like symptoms. And then we could look directly for the alpha-synuclein protein and ask whether or not it was more likely to be in the amyloid-like form if the mice were colonized with curlated bacteria. And in fact, it was. So it was in this protease-resistant punctate form in the mice that were that were given the bacteria that had curli, and then it was still mostly in a soluble-like form, protease-sensitive form, in the mice that were given the bacteria that could not make curli. So then the question was, well, could CSGA directly cross-seed or accelerate the alpha-synuclein aggregation? And so if you look at alpha-synuclein aggregation, as Ehud pointed out, I think that it's relatively slow in a test tube. So in a test tube, alpha-synuclein, it will get into an amyloid-like fiber. So this is that same THIO-T assay. It'll get into an amyloid-like fiber, but it takes it quite a while. And on the order of 10 to 15 hours of a lag phase before it gets into an amyloid. But if you spike in just a little bit, in this case, about 1 to 20 or so amount of the CSGA protein, now you can see that amyloid formation has quite sped up. So this is all alpha-synuclein aggregation here. We're not looking at the CSGA aggregation in this case. We're just looking at alpha-synuclein. And you can see that we've decreased the lag phase by about 50% or so. E. coli, in original studies, it's a little bit hard to know, but E. coli don't seem to be overrepresented in Parkinson's microbiomes. That's where the CSGA protein comes from. And so we asked if there are other bacterial CSGA-like proteins present in the human microbiome. And this was a project that was spearheaded by Sujit in the lab. And so what Sujit did was he took what we knew about the CSGA protein from E. coli. It contained these 5 imperfect repeating units, and we were able to sort of design some programs to pick out the what we knew were the amylogenic regions of these repeating units, and then we could use those to screen through the microbiome databases and pull out CSGA-like homologs. And it turns out there was a lot of these homologs. We only pulled out a few of these and characterized them. But I'm going to show you a couple different examples. An example from a Hafnia CSGA-like homolog and a Citrobacter homolog. And I'm using these two because they were sort of the extremes. So first of all, Sujit was really good at picking out amylogenic proteins. The ones that we picked out and that he cloned and characterized, they did form amyloid in vitro. They had different properties. The Hafnia, for example, formed amyloid quite slowly. So you've got a long lag phase and then it forms fibers, but it forms very amyloid-like fibers by TEM and by beta sheet rich by circular dichroism and things. And then the Citrobacter CSGA homolog that we pulled out, this one had almost no lag phase. It's very hard to measure the lag phase on this one. It gets into an amyloid quite quickly and forms fibers again itself, but it does so quite fast. So you could characterize these by the length of their lag phase. The E. coli was sort of in the middle of these two, where Hafnia had a really long lag phase and Citrobacter had quite a short lag phase. And then we looked as to whether or not these could stimulate alpha-synuclein or accelerate seed alpha-synuclein aggregation in the same way. And the Hafnia did. So Hafnia, here's alpha-synuclein in the blue line. And then if you spike in just a little bit of the Hafnia protein, it accelerates, cuts that lag phase in half, just like the CSGA from E. coli did. And the Citrobacter CSGA didn't have as much of an effect on alpha-synuclein. So it was accelerated just a little bit, but not very much. It wasn't enough that we could make too much of. And at first we just thought, oh, well, just not everything accelerates alpha-synuclein. That's probably good. But then Sujit sort of had this thought, that, well, maybe it's the intrinsic amyloid forming propensity of these CSGA homologs that govern their ability to accelerate alpha-synuclein aggregation. And so maybe the CSGAs that get into an amyloid very quickly themselves don't cross-seed as well as ones that spend more time in that lag phase. And then he thought of a way to test that. In order to test that, I need to back up and tell you about a really old paper from the lab that Xuan Wang did in our lab years ago. And this was with the E. coli protein of CSGA. So Xuan was studying CSGA, and he was trying to understand what was important for CSGA amyloid formation, and he was making all these mutants. And he made a few mutants that actually made the protein accelerate or form amyloid faster. And this was interesting to us because he took a naturally forming protein that was supposed to get into the amyloid form, and yet was able to make a few mutations, specifically in these aspartic acid residues. There were 3 really important aspartic acid residues, but there were a couple others that played a role for sure. These aspartic acid residues that if he mutated them, he could mutate them to any amino acid, it seemed like, except for a negatively charged amino acid. And when he did so, he got a CSGA that formed fibers faster. An interesting side note is that these were really toxic to the cell. So they were really tough to express and tough to get out of the cell. So obviously nature has figured out just the right level of amyloidogenic form for the CSGA protein. But anyway, what Sujit noticed, which I think was really clever of him, because I never would have noticed this, is that the Citrobacter protein that we had pulled out, it forms amyloid really fast. He noticed it was missing those gatekeeper residues. Those gatekeeper residues turned out to be really conserved in most of the CSGA homologs that we could find, except they were not in Citrobacter. They were changed to other amino acids. And so Sujit added those gatekeeper residues back in the same positions as they were in E. coli, and he made a protein that was had a much longer lag phase than the wild type version of the Citrobacter CSGA. And so this is interesting. So now he's taken this protein and he's added these gatekeepers back, and he's got a longer lag phase here. And now he can test his hypothesis. Does this accelerate alpha-synuclein aggregation? And in fact, it does. So here's alpha-synuclein aggregation in the blue line. And then if you add this gatekeeper version of the Citrobacter protein, it decreases that lag phase about in half again. And so it suggests that some pre-fiber species maybe is what's accelerating alpha-synuclein aggregation. And just to kind of connect the dots, that is what E. coli secretes during the assembly of curli. It secretes a pre-fiber version, an unfolded version of CSGA, and we think that could be interacting with alpha-synuclein or other proteins in the environment. A question an ongoing question that I'm not going to spend very much time on, just so that you can all go home and go to sleep if you're living over in Europe and other places. So can we intervene with interactions between the functional and disease-associated amyloids? And we've been trying to do this for a long time, and I'm just going to be general here. We've used protein inhibitors of these process proteins that come from the bacterial systems to inhibit amyloid formation or inhibit the interaction between CSGA and alpha-synuclein or other proteins. Here's a human protein, TTR, that you might be familiar with. This protein in its monomeric state very much inhibits both alpha-synuclein and CSGA amyloid formation and the accelerated amyloid formation that we see when we add the two proteins together. And then with the Almquist lab and others, we've been working to identify small molecules that can also interrogate this process and block amyloid formation. And then as microbiologists, we're really interested in this biofilm formation and how to sort of interrogate that side of what the bacteria are producing these amyloids for. And so just to summarize, I could read through all this. The first thing is the most important thing, because others have shown in really beautiful papers from Friedland and Zhang and colleagues more recently have shown in other model systems. So we did our initial experiments in a mouse model system, and they have shown in a C. elegans model system and a rat model system, the same interaction and the same ability of curlated bacteria to make alpha-synuclein or drive alpha-synuclein into that aggregated toxic form that's associated with Parkinson's disease. And I'll not read through all of these just so that I can get to the most important thing, which is the people that did the work. I'm just foreverly grateful for the people that are currently in my lab, or mostly currently in my lab. There's a couple that have just moved on from this picture, but the people that did a lot of the work. So Sujit is right here. He did a lot of the work that I talked about. Neha Jain, who was a former postdoc and now runs her own lab in India. She did a lot of the initial sort of discoveries many years ago that curli in a test tube anyway could accelerate alpha-synuclein aggregation, and that really spearhead a lot of our thoughts going forward for this stuff. And then our collaborators, who I'm grateful for, all up and down the list. I mentioned Frederick Almquist and a couple of other ones, but yeah, and the funding agencies that have helped. So thank you very much for listening and staying awake if it's late in your town.
Thank you so much, Matt. Wonderful, amazing that all this connects, right? That the gut is so important. And there you have the amyloids too. Let's see, we don't have any questions yet, and maybe we should not stay too long, but I want to ask a few things. And I think my fellow co-speakers here, we can all ask now, right? 5 minutes. Oh,
No,
yeah. Oh, you want to ask? Yeah.
I do. So what I'm struck by in all of this, right, is that we're increasingly recognizing different entities that can assemble and that they can be polymorphic and also interacting. So as we probe deeper, clearly there's so much complexity here, right? And I guess as a speculative question, I'm wondering to all of you is, how common will it be, I wonder, that we could have multiple entities causing each other to aggregate, but actually by interacting. So one that I was thinking of was A beta and tau. There's also now evidence that they're, this is why the simple strategies at targeting aggregation are failing, is because those are actually doing something together. And, as you were showing now with curli and alpha-synuclein and with, the Rim proteins, and I wonder even with these metabolites that Ehud has, and, there are so many possibilities here, I think. And I wonder, how big is it going to be, right? It's as you said, Matt, right? There's this, we used to think it was the exception. Now things are the rule. So I'm wondering what you all think.
Yeah, I think that's a really interesting question. And I think, 10 or 20 years ago, I think the dogma was that seeding and the interactions between different amyloid or amyloidogenic proteins was very specific. And we saw that, especially in some cases it is, in the prion field, that was, very clear that you could only get certain strains or certain variants, to cross seed or to cross, to interact with each other. But I think as we discover more and more of these aggregation-prone proteins that end up in this very, conserved structure, as we discover more and more of these, I think we also recognize that there's a lot of promiscuity between I think some people have mentioned it in the question and answers before, between the actual structures that those proteins can get into themselves, that maybe there's some heterogeneity in the structures that maybe CSGA could adopt. Maybe it could adopt a slightly different structure depending on who it's around or what it's interacting with. And the same may be true of these of some of these other proteins. We know, for example, that the curli proteins, we've known for a long time that these curli proteins can be very different. The CSGA proteins, the homologs that we pull out, they can be quite different sequence-wise, quite different, but they will almost always be able to cross-seed with each other and be complemented with each other. And we've thought, well, maybe that's a good way for bacteria in a heterogeneous biofilm to be able to, use subunits from another species or to make connections with another species. But certainly it brings up the possibility that maybe these functional amyloids are a little bit more, sometimes could be heterogeneous, sometimes could not. And maybe those interactions would fall from that too. I think
And
we're
they
just
have
at the
been
very beginning of understanding that.
Yeah, because I mean, you can add to that, like just taking one protein like synuclein, and the structures depends on the conditions, right, of the amyloids, and if it's from a patient or from a test tube. So there seems to be a lot of variability, and I think here all the structures that are coming out. I mean, there's a lot of amyloid structures coming now, and it's almost overwhelming because we see so many folds. But eventually, that will help us, see what's possible, maybe.
Yeah, and that's a
But
good
it
point
feels like
too,
it's very
because
generic, right? It's like,
yeah, because it's only been in the last 20 years or less that we've had any structures, right, of the amyloids. It's very general. We knew they were beta sheet rich and cross beta strand, but then it was like, oh, well, what is it? We don't know. And it's only been in the last, 20 or so years that we've really gotten good structures, whether it be through solid state NMR or through the great, crystallography that Eisenberg and his colleagues have pioneered, the microcrystals.
Kind of a young field. It's a little bit like IDPs, or intrinsically disordered proteins, that before we thought every protein was folded, right? Now we know it's actually a large amount of proteins that are not folded. And now we're starting to learn that a lot of proteins can get into amyloids in all kinds of ways, right? And that's functional.
Yeah.
Oh, we have a question, Matt, from Nia. Hi, Matt. Nia here.
Oh my gosh, I didn't know she was there.
Yeah, don't. She asked the question before, I think. Yeah. Do you think Curly would cross the blood-brain barrier and can reach the brain?
Yeah, Neha, yeah, of course, that's a great question.
Yeah, and I wanted to ask that too, so that's good.
Yeah, I wish I had a definitive answer for that. Of course, it's the question, I think. Where would this interaction be happening? Number one, in the animal models, where we've looked at it, or if this occurs in humans, where would it be happening? It could happen in the gut. It could happen in that vagal nerve, that vagus nerve that I talked about before. So it wouldn't. So there could be some interactions there, that could be propagated then to the brain. But yeah, we're a little bit weak on that, Neha, as you might know. We don't know for sure. It's a great question.
Do you
Certainly,
know,
the blood-brain barrier becomes compromised under some of these conditions, and so.
And the gut also, right? But do you know CSGA can go into like enter endocrine cells or?
Yes, it can. So that's known. Yeah. So it can.
And I mean, synuclein is around there, right? So you can
and it's
meet
there,
and you
right there.
get a seed and then you go. Yeah.
Yep. We just haven't caught it, in those animal models. We just haven't caught those initial interactions.
I would say there's also, there's a lot of work now, I think, happening around viral infection and the potential for viral proteins to Z-day beta in the brain. It is a bit about, as you said, right place and right time and concentration. We sort of started with amyloid thinking, well, it's a generic beta structure. Now, and then we moved all over, oh no, the sequences are amazingly important. If you look at the structures, the side chains are critical. And now perhaps we're backing off to say, well, there's a place where incompatibility of side chains is tolerated to some extent, isn't it? And that's where you'll see the heteromeric seeding. But you still need those two potentially compatible proteins to be in the same place at certain concentrations. And I think we're grateful for that. Otherwise, we'd all have a lot more problems.
Yeah. It's all about location and timing. And then if everything was everywhere at the same time, all kinds of things could happen.
That's
but
right.
It's kind of interesting, the COVID story. I mean, in principle, the studies that tend to show that maybe some parts of the COVID proteins can trigger Parkinson's, Alzheimer's, and that's maybe something we know in 5-10 years.
We haven't.
Yeah, I think the inflammatory response and that response could absolutely contribute to these, because you would you obviously change protein folding with those responses. So
inflammatory response we didn't discuss at all serum amyloid, and
Yeah.
it's
Yeah, exactly.
Yeah.
islet amyloid polypeptide, type 2 diabetes. Some people call Alzheimer's disease type 3 diabetes.
Type 3, yeah, exactly.
Yeah, how insulin degrading enzyme mutation actually cause Alzheimer's disease. This is all much. It's only the tip of the iceberg in my view.
Yeah, I agree, Ehud. Yeah, agree.
It's so much more. We can probably do many more webinars.
Yeah.
Not us, but others that know their thing.
Others.
Yeah.
There's one question here that Matt may answer, but actually Nea and I may answer because she's coming to my lab next year.
Oh, nice.
Can bacterial amyloids seed animal and fungal amyloids, even if they have different sequences but similar beta sheet structures?
Yeah, well, in the case of, so we've tried to look for CSGA anyway. We tried to look at quite a few of, what it might be interacting with, and really the only thing in my lab now. Others have suggested that maybe A beta could, or CSGA could also accelerate A beta aggregation. So Maida Landau's lab has shown that quite nicely. For us, we have only been able to show that CSGA of the human proteins anyway, really does anything to alpha-synuclein. And it was really just alpha-synuclein that we could see, sort of clear differences in a test tube anyway. And so that was, yeah. But a lack of a result is not an answer in this case. I think the answer is yes. I think there probably are other interactions that are happening between these bacterial ones and maybe the fungal proteins too.
We're going to have a look.
Yeah, exactly.
Well, I think this has been a fabulous discussion, like fabulous presentations and discussion. I think I'd just like to thank you all again. This is really exciting, and I agree that we're kind of going to learn so much more. The tip of the iceberg, as you say. So thank you, everyone. Thank you, Pernilla, for leading us on this, and Matt and Ehud and Margie. This was really great.
Thank you so much for to everybody, and to you coming in really, really late, really early. And great time for you, Matt. But that's
Margie, go take a nap.
Already at the beginning of the day. Now it's
go to bed, Ehud.
I'm fired up. I'm fired up for the day. It was really great.
Yeah,
so
now you get going, right?
You hear your presentations. You've sparked lots of ideas for me too. So thank you. Lovely to see you, and take care, everybody.
Thanks so much.
Thank you to everybody out there too.
Yeah, lovely to see you.
Everyone.
And thanks for Luca too. Bye.
Bye.